Hey team, this is your chemistry coach coming at you with our first lab video. This is for lab number 9 on kinetics. I'm just going to do a quick little pre-lab. I don't do a lot of pre-lab talks for you guys at Chem 185. You can read your own stuff, but it's a pretty crazy pre-lab for this one. So I wanted to do a quick little overview. I was going to do this in lab anyway, but we can't meet in lab, so we're meeting in lab right now, right? So my hope is to go video the results for you, post that as a video on Canvas. If you look under labs in Canvas, I'm going to put some folders. You'll see like the kinetics lab, activation energy lab. Not a lot of skills in this one. We just mix some stuff and watch it turn color. So anybody could do that. So I'm just going to video myself doing that and showing you what the data is. And then what I'm going to do is have a data sheet that you can print out and fill in the data based on the video. And you can rewind the video if you missed some of the data. And then there's also going to be a calculation sheet. So you have this pre-lab video. You're going to have the lab video itself with all the data sheet you record all your information in, and then the calculation sheet with all the results, and a nice little graph and plot you're going to make. And you're going to send those in to me, the data sheet and the results and calculation sheet. And I'll just grade you off of that. It's going to be a lot of calculations. That'll keep you busy over spring break. So hopefully in the next couple of days, I'll be able to get in on campus. Hopefully they'll let me in so I can run this experiment and have it recorded. So let's talk about this experiment. All right. Our goal is to figure out the rate law for a reaction. That's reaction number 1. That's the reaction of interest. So some iodide ion reacting with, that's a new one for you. That's peroxydisulfate. It's like 2 sulfate anions connected together. So it's peroxydisulfate. Peroxy meaning they're connected by an oxygen bridge. So we want to know the rate law for that. It's a slow reaction. It's going to produce the triiodide ion, which actually has a yellow color to it. I don't have a yellow marker, but that is a yellow color to it. And we're going to get some sulfate anions over there. We're going to use a twist off the method of initial rates for this, which means we're going to hold one of them constant. And our goal in this lab is to hold the I minus constant. And there's an interesting little trick we're going to do that. Thus we have reactions 2 and 3. We're going to use those to hold the I minus constant. So what we're going to do is we're going to vary the concentration of the peroxydisulfate anion and see how long. We're going to do 6 different reactions of 6 different concentrations. So what you would have done in lab is made 6 different flasks with different concentrations of peroxydisulfate ion. I'm going to do that for you. We're going to mix it with a constant concentration of I minus and see how long those take for a reaction to occur. And we'll be looking for a color change. We'll time those, right? So we'll know the change in concentration and the change in time. We can calculate rates and stuff. And I'll do that after we talk about the basics of what's happening. I want you to understand the process of the reactions. How are we going to hold I minus constant? All right. So what we're going to do is we're going to add some thiosulfate. That's what this is. So this is thiosulfate ion. This was peroxydisulfate. That's a long one. So peroxydisulfate ion and the thiosulfate. It's like a sulfate ion, and you replace one of the oxygens with a sulfur. That's all. So thio is a sulfur term. So we're going to add some thiosulfate of a low concentration in our experiment. This is a very fast reaction. So the triiodide ion that we're producing in step 1 will immediately react with any of the thiosulfate that we've added to replenish the I minus. Look at this. So that I minus forms and gets recycled back. So that's going to hold the I minus constant. So as long as there's thiosulfate ion present, the I3 minus cannot build up. It goes bye-bye immediately because second reaction's fast, regenerates I minus. So I minus will stay constant as long as you have thiosulfate. But we're purposely going to make I'm going to show you a graph in a second. We're purposely going to make the thiosulfate a lower concentration than everything else so that it goes to 0 first. When that goes to 0, we can no longer run reaction 2. What happens is we go to reaction 3. So the I3 minus no longer can react with the thiosulfate because it's gone. It'll react with a starch complex we're going to add. This is where it's cool. When you take the triiodide ion with sulfate, you get this really, really dark blue complex. It's a really fun to see. And that's what I'm hoping to record, where you can see that really cool blue color change. So we're going to mix these two solutions, right? We're going to, inside our I- solution, we're going to add the thiosulfate and the starch. And then we're going to mix that with a known amount of the peroxide sulfate, mix them together, call that time 0. When it turns blue, that's time final. We can calculate delta T from that. It's pretty easy. So once that runs to 0, it turns blue and the reaction is done. So what I'm going to do is I'm going to put up a graph that shows you how these concentrations are changing over time and how we can calculate the rates and get the rate law from that. So this is in your pre-lab. Look up and read through your pre-lab for the kinetics. This is lab number 9, kinetics. All right, guys, what we're going to do is we're going to make a graph here and show you what's happening with the concentrations of these 3 main species over time. We're not worried about the starch. That's just present at the end. So these are our main reactants, the I- and peroxide disulfate, and this is the thiosulfate that's supposed to go to 0. So over time, this is going to drop, right? This is going to drop. But remember, the I- is recycled. As long as there's thiosulfate ion in there in reaction 2 on the prior slide, the prior board, the I- gets regenerated, so the I- stays constant over time, right? As long as this is present. But because we're making a lower initial concentration of the thiosulfate and the peroxide disulfate, this will go to 0 first, and that's when it turns blue. And we're going to call that T final. Right? This will be T initial. Now, these will be the initial concentrations. You see the sub-0 means time 0. You could put a sub-0 or you could put an I for initial. You'll see sub-I or sub-0. Doesn't matter. It means the same thing. And this will be the final. Now here will be the final concentration of your thiosulfate, which obviously is 0. So that one's easy. So we could easily calculate the change in this, right? So the change in the thiosulfate will just be the final minus the initial. O32 minus final minus S2O32 minus initial, right? Or that could be a sub-I there. So at that time that it turns red, I dropped my pen, at the time that it turns blue, this will keep dropping, right? But this will stay constant. And let's draw a little vertical line here at this point. Right at that point, the thiosulfate runs out, which means the I- then starts to drop. So notice the reaction of interest is still going after it turns blue. So we're stopping it here because we can't have the I- dropping. It has to stay constant. So we have to end it right there. Right? We can also look at this as the final I-, right? So this will be I- final, which should be the same as the initial, right? This will be the final S2O82 minus, which we can't solve. So that one we can't solve for. We don't know what it is. It's up there, right? We're going to be using what's called the average S2O82 minus. So we're going to be using a because that changes over time. So we're going to be using an average value. And in your pre-lab, it shows you how to calculate that. There's a cool little equation that shows you how to do that. So we can calculate the change in any concentration as long as we know the initial and final, but we only know that for the I- and the S2O3. We can't do that for S2O8. So there's a little trick we're going to do to calculate the average. So what I'm going to do now is write up the rate law, show you how we're going to manipulate that with our logarithm tricks. Remember your stupid log tricks, to show you how we're going to calculate. We're going to make a plot and calculate the exponents with the species. So let's look at the rate law real quick. All right, what I did is I rewrote reaction 1, which is the reaction of interest, the iodide ion reacting with the peroxide disulfate, and our goal is to figure out the rate law for that. So let's write the rate law. So the rate will be the rate constant, remember, times the concentration of reactants raised to some exponent. So that's going to be the concentration of I minus raised to some exponent. In lecture, I used N and M. In the lab, they use A and B. Who cares? X, Y, A, B, whatever you use, doesn't matter. Times the other reactant raised to the power of B. And that's what we're after here. So once we use our data, we're going to be able to solve for B. And once we get B, I'm just going to give you A, right? So there's actually two parts to this. One where we figure this one out by holding this constant, and the other one where we figure this out by holding this constant, but it's the same lab. So I'm just going to do the first type of lab. We're going to vary the S2O8, holding I minus constant, and from a graph, we're going to figure out what B is. And then I'll just give you A, and we've got the rate law. We'll be able to solve for the rate constant as well as we're doing that. Now remember, rate we can do with any species in there. So I'm going to write the rate as the loss. Remember, reactants are negative, products are positive, and it's 1 over the coefficient. Since the I minus is constant, I'm going to focus on the S2O8. So that will be the negative of the change in the S2O8 2 minus over the change in time. So very easy, if you look at the graph we did, you could get the change in time, right? The time it turned blue versus the time you mixed them. And the change in the S2O8 2 minus, you just take the final minus the initial, but we don't know the final. We're not going to be able to do that. So we're going to get the change in the S2O8 2 minus, if you look in your Prelab, from the change in the S2O3 2 minus, because we do know the final of that one, which is 0. So we're going around through the back door to figure out the change in the S2O8, but we can calculate both of those. We can calculate the rate. So now let's apply some tricks to this rate law. You ready? I'm going to take the logarithm of both sides. Let's do this. Follow me. We'll do the natural log. So the natural log of the rate, and would you agree that the rate constant's a constant, right? The I minus is a constant, and the A will be a constant. So I'm going to call all of this right here, right there, C1. I'm just going to lump all those constants together and just call it constant C1. We can call it anything we want. So I'm going to take the log of both sides. So the log of the rate will be the log of that constant times the S2O8 2 minus raised to the power of B. Now, do you remember stupid log trick? Log of A times B is log of A plus log of B. Do you remember that little trick over there? So I'm going to split this from this, right? So I'm taking the log of this whole thing here. So I'm going to split it and say the log of the rate will be the natural log of C1 plus the natural log of the peroxide disulfate concentration raised to the power of B. So I'm using this log trick to separate those two. Boop! Gotta love logarithms. They rock. Now remember another stupid log trick. Log of A to the X power, you can move that out front and go X log of A. You see this exponent here? I can just move that out in front of the logarithm. Remember, I'm trying to solve for B, and I'm isolating B using stupid log tricks. So let's do it. And then I'm going to flip these two. So the log of the rate will be B times the natural log of the peroxide disulfate ion concentration plus the log of that constant. That's the equation we're after. Does that look familiar to you? Like y equals mx plus b. How many times have we done this already this semester? So in this scenario, y is the log of the rate. So we're going to plot that on the y-axis. The slope will be equal to the exponent with respect to the S2O8, which is what we're trying to solve for. So we get the slope of this graph. Boom, that's B. X will be the log of the S2082 minus, and we're going to use the average concentration for that. And then the y-intercept will be the natural log of C1. Now from the y-intercept, we'll be able to calculate C1, and from that, the rate constant. That'll be the last thing we do for this lab. So we're going to make a graph. Let's show you what this graph looks like. If we plot the log of the rate on the y-axis versus the log of the average S2082 minus on the x-axis, we're going to get a least squares fit, giving us a slope. Let me put that up on the board. All right, so if we do this graph, log of the rate versus the log of the average peroxidized sulfate ion concentration, and do a least squares fit, which you're going to do on your Microsoft Excel. So you'll be turning this graph in along with your data sheet and your calculation sheet, and I'll be grading those. It's going to be a lot of calculations, which we'll talk about in a second. Now remember, the rate was the negative of delta S2082 minus over delta t. So we can calculate rates and take the natural logarithms. This you're also going to get. So from this, our slope is equal to b, which is what we're after from the rate law, correct? So let's draw that rate law again. Is k I minus to the a power S2082 minus to the b, and that is what we're after, gang. So all you're going to do is get a slope, and that'll give you some integer like a 0 or a 1 or a 2, right? 0 order, first order, second order, third order with respect to that species. And once you have that, I'll give you the A. Pretty straightforward, but the calculations are nuts. So let me show you real quick how you're going to do some calculations, and then when I can video the actual results, we'll put that on as a video, and you can extract your data from the video. So let me show you how we're going to mix these solutions called a double dilution. All right, so what we're going to do, and this will be on the video, so this shows you how to do the mathematics for the video. Lots of redundant calculations, because you'll have 6 different flasks of 6 different concentrations of S2082 minus. So the calculations are the same for each flask. It's just, it's different. You got to do it for each flask. So you're going to have pages and pages and pages of redundant calculations. But once you do it for the first flask, it's exactly the same for the rest of them. You're just starting with a different starting concentration of that. So in lab, what I'm going to do is make up what you would have done is make up 6 different Erlenmeyer flasks with 6 different known concentrations, right? So you're going to mix some water with, so I'm going to make an original peroxidized sulfate solution with a concentration, known concentration. We're going to dilute it with varying amounts of water to create 6 diluted concentrations. So we're going to have to use the M1V1 equals M2V2, or the C1V1 equals C2V2. So this is your dilution equation, right? So you're going to have to calculate the concentration of your peroxidized sulfate ion concentrations in your Erlenmeyer flasks, 6 different ones, using the dilution equation and the data I provide. So a certain volume of this, certain volume of water, boom, you mix them together. You're also going to make another solution in a giant 500 milliliter graduated cylinder. You're going to add some iodide ion from potassium iodide solution, some thiosulfate from sodium thiosulfate solution, and some starch solution. So you're going to have known volumes and concentrations of those added in here, and then we're going to dilute it up with some DI water up to around 316-ish milliliters. I'll let you know what that is in lab. So we'll be able to calculate the concentration of all of these using the dilution equation. So we'll know the concentration of everything in this big cylinder. We'll know the concentration of everything in these 6 different Erlenmeyer flasks. And then what we're going to do is we're going to take 50.0 milliliters of this iodide starch thiosulfate solution and add it to each of the 6 flasks. That's why we have a little over 300 milliliters here. So 6 flasks times 50 mils, we need a little over 300 milliliters. So we're going to line up these 6 Erlenmeyer flasks of different concentrations and about 50 milliliters and go boom, 50.0 milliliters of this, boom, 50.0 milliliters of this into the second flask, boom, 50.0 milliliters of this into the third flask, fourth, fifth, sixth flask, swirl them, boom, that's time 0, and pick our nose until they turn blue. Hey, that's the final time, and you're done. That's all the experiment really is. But we're going to be doing what's called a double dilution. Double dilution. We're taking our original solutions and diluting them twice. The first dilution is making this solution here and this solution here. So you're going to use the M1V1 equals M2V2 to calculate those starting concentrations. Then the mixing process dilutes them again, because we're going to take 50.0 milliliters of this, of a starting concentration for each of these species, mix it with around 50 milliliters of this, that species. That dilutes it because the total final volume will be the volume of the peroxide disulfate plus the 50.0 milliliters of the iodide thiosulfate starch solution. So you're going to do the M1V1 equals M2V2 twice for the I minus, twice for the thiosulfate, twice for the peroxide disulfate, and that will give you those initial concentrations just after you mix them that I showed you on that graph, that starting concentration on the graph, and then they all go down except the I minus, which just stays constant, and the S2O3 goes to 0. That's going to be a lot of fun, all right? So I'm going to leave this as is, and you can study this and the pre-lab itself, and when I get that video, I will post it. It's going to be a very simple lab to run, but some intense calculations. It's going to take you most of spring break to do it. Yay, something to do.